Started. All right. So again, thank you all for joining us today. Welcome to the Wolfpack Wellness web series on wellness research. I am the host, Suzie Goodell, an associate professor in nutrition in the Department of Food, Bioprocessing and Nutrition Sciences in the College of Agriculture and Life Sciences. Whew, that's a big name, right? As a reminder, the Wolfpack Wellness web series is a collaboration between the University Wellness Advisory Committee, the NC State Libraries, and NC State WellRec. This series aims to discuss the wellness research happening across NC State's campus. And just to remind you of this, that there are so many different types of wellness, including physical, emotional, social, purpose, community, and financial wellness. We will be exploring all different types of wellness in the NC State research throughout each show. And today's show is with Dr. Randy Jirtle. Now, I will say that we will have a wonderful time to do Q&A at the end of the show. So if you have questions as we go along, please feel free to type them in the chat, and our lovely intern Kendall will manage those at the end. So just to give you an introduction, Dr. Jirtle is presently professor of epigenetics in the Department of Biological Sciences here at NC State. His early studies were in the field of liver carcinogenesis, which led to his identification of the first imprinted tumor suppressor gene and the determination that the phenomenon of genetic imprinting evolved in mammals about 150 million years ago. Dr. Jirtle is the first, provided the first molecular evidence. He's not the first molecular evidence. He provided the first molecular evidence that—
I'm old, but not that old.
Exactly. That maternal dietary supplementation of agouti mice altered the coat color distribution and the disease susceptibility of genetically identical offspring. Those are lots of terms that he will help us understand more about. Dr. Jirtle has received many awards in recognition of his achievements, and his research has also been featured in a PBS Nova television program on epigenetics entitled Ghost in Your Genes, and a Shortcuts TV documentary entitled Are You What Your Mother Ate? The Agouti Mouse Study. So just that alone, he's like, I huge crush on that one. That's amazing to be part of PBS Nova in my personal.
It is pretty amazing, isn't it?
Right.
I mean, I never even thought I used to watch it always. And the think about being in it it's really amazing. It was incredible. I loved it.
Well, welcome, Dr. Jirtle. We are so thankful for you joining us today. And, I said I used a lot of big terms when introducing you, but I'd love for you to tell us a little bit about yourself and your research lab.
Well, when you open it up that way, I can talk forever, I mean, I originally came from Wisconsin, I'm now back in my home state, and got my undergraduate and graduate degrees here at the University of Wisconsin. And fortunately, up until just a few weeks, literally a few weeks ago, I was also an adjunct professor here at the University of Wisconsin in the Cancer Center. So my whole sort of career has kind of gone full circle. But I was at Duke University for 35 years, starting in 1977 and retired in 2011. And then the people that I was interacting with, which one of the main ones was Catherine Hoyle. And then after I retired, she then went to NC State. So I don't have a research laboratory at NC State. She works in my lab at Duke, and now I work in her lab at NC State, but it's more in an advisory role and reading grant applications, writing papers, that kind of stuff, rather than actually running the lab, because Catherine runs the lab with David Scarr, who was a postdoc in my laboratory. He then also went over. So those are the two now that are at NC State. So if anybody's interested in the field of human epigenetics and epidemiology, get in contact with David Scarr and Catherine Hoyle, because I can't do anything like that anymore. I don't run the laboratory. Yeah, so I mean, I think I'm going to stop there, Suzie, because that kind of gives a broad background of how I got to Duke, where I came from and stuff like that. I did not start in the field of epigenetics. I'm going to add one more thing. My undergraduate degree here at Wisconsin, because I'm here in Madison right now, was in nuclear engineering, not even in biology.
And I think that's a really great example of how we can shift where we are headed if we are open to where the science takes us, or the
crazy.
or the grant funding. So we're going to talk specifically today about your work in epigenetics and the imprintome. That's a word I just learned today. So I'd love to hear more about what spurred your studying of this kind of concept, and tell people out there what epigenetics is, because I don't know that a whole lot of people know that term.
Yeah, the term epigenetics was coined quite a while ago, actually, but it literally, epi just means above. So we're just talking above the genetics. Now, that doesn't tell anybody very much about what it really means, but I like to think of it this way, because this is the way it actually really does work in cells, that the genome is comparable to the hardware of your computer, the physical machine, the chips and all the keyboard and screen and stuff like that, but the chips in it basically. And the epigenome is the software that tells that computer when, where, and how to work. So you can view a cell from an engineering standpoint now simply as being a programmable computer. So everything you were talking about, all these environmental effects, ultimately if they're going to have an effect, those effects have to be transduced down into the DNA and the histones that the DNA is wrapped around and change programs. So while we can have 260 different cell types in our body, but every cell has the same DNA, is because every cell type is literally running a different program, which was established during their very earliest stages of embryogenesis. It's beautiful. So I'm going back now, if you think about it, from my simple way of looking at life, I always loved computers and programming, even way back when I was here first as an undergraduate. I took a number of courses in computer programming. So in effect, what I'm in right now, and I felt like I came back home in biology when I got into the field of epigenetics, because basically we're talking about chemical programming now that changes the way genes are expressed in our whole epigenome, or our whole genome, basically.
So tell me a little bit about how this whole world of epigenetics plays a very critical role in health and wellness.
Well, I mean, in the old days, and it's not that long ago, I get back to epigenetics now. Epigenetics initially is studied primarily in cancer formation. And then there was another field in the late, it started in the 80s and went into the 90s. That was its beginnings of genomic imprinting. And we'll get into that later on as you ask. This is the imprintome stuff. So there were sort of two groups of people that were working in the field of epigenetics. But the important thing is that when anything impinges upon the cell, and particularly it can, let's think of it as damaging now the cell, let's say, you can damage two really important components, just like in your computer. You can damage the hardware of the computer. In other words, your hard disk goes out, or literally one of the chips in your computer goes bad. That's the genome, mutations, deletions, that kind of thing. So environmental effects can do this. But the other thing that it can do, which was never really thought about very much, but it has to be thought about, because if you don't look at both of them, you're never going to really know how to fix the computer. And that is, it can also, and often does, alter the software, bugs. So if anybody's used computers, which many people do now, they know that many times their problems are not hardware problems when it doesn't, quote, work. I mean, the problem when the pipeline was shut down was not a problem in the hardware. It was a problem in the software. And it's the same thing in our cells. So health and well-being is not just dependent upon having mutations not occurring, or if they do occur appropriately, repaired in the genome, but also having those programs appropriately running in the computer itself, because in both situations, it can wreak havoc on the way the cell functions and causes diseases.
So tell me a little bit about, and thank you very much for that answer, because I think that really brought it home to a lot of us who aren't in the world of cellular molecular genetics.
It's nice to have something to hang it on.
Yeah.
And it's very simple. I mean, the processes are not so simple, but the concept is very simple.
Yeah, it's a nice analogy. So tell me a little bit about how, when you approach a problem related to epigenetics, can you give an example of something that you've done? I know we were talking about agouti mice and how that might translate, but are there things that really stand out in your mind of the work that you've done, or maybe you're even currently doing, that can give people an example of where epigenetics really relates to the everyday person?
Let me say that that's a huge question, unfortunately. To get into this question, we've got to talk a little bit, I think, more about genomic imprinting and how I got ultimately into the agouti mouse study.
So why don't you tell me a little bit about genomic imprinting?
I will do that. Genomic imprinting is a very simple concept, but very weird. So these are autosomal genes that are regulated in a very unusual manner. They're expressed in a parent of origin dependent manner, and only one copy is expressed. So in other words, go ahead.
Oh, wait, I think you might have told me that you were talking about there's two genes, but only one of them.
There's two copies of every gene because they're autosomal, mother and father. Mother brings in one, father brings in one. Normally when most genes are expressed, they give rise to RNA and protein. Both copies work. And if they're shut down, both copies are usually shut down. There's a subset of genes of which only one copy works. The other one is silenced epigenetically. And that silencing is parent of origin dependent. And it's dependent upon regions that are close to the gene that control that imprinting regulation. So every imprinted gene will have a regulatory element that shuts down the mother's or the father's, copy, depending on the gene. And we have 500 to 1,000 of those spread throughout our genome. The imprintome that you're going to be talking about is the accumulation genome-wide of all those regulatory elements that control the phenomena of genomic imprinting. These are the targets basically for environmental insults screwing up the expression of an imprinted gene. Unfortunately, from our standpoint, really, is that it looks like we got here literally on the backs of imprinted genes, but now we're vulnerable because we don't have a backup on these genes because one copy is not functional at all. So now if you have a mutation in the only gene that you inherit, let's say from your father, and it's not functional because of the mutation, the mother's copy could be completely fine, but it's not working. So you see immediately the problem. This is a dominant effect, a mutation, it shows it. And you can not only have problems now because of physical genetic mutations, but epigenetically, if you silence, now you have no expression. Or if you inappropriately loosen up the expression of the silence gene, now you've got double the dose, and that is also problematic because these genes tend to be growth regulatory and they wreak havoc in development. So for appropriate development to occur, we need only one of these genes and they have to be functional. It's incredible that we evolved these because it makes us so vulnerable.
So when you talk about it's really important in growth and development, is this most important in embryonic development or fetal development, or does this really also apply outside of the womb?
Well, it does once the individual is born. There's two places where imprinted gene expression occurs. The monoallelic expressed genes are expressed most commonly in the placenta, controlling the ability of the fetus to in effect bring nutrients into its body as it grows. And the other place, the brain. So once the individual is born, it doesn't have to worry about the placenta, obviously, but if there's not appropriate expression of the imprinted genes during brain development, it gives rise to what we call behavioral disorders, all of them.
So how do you work with epigenetics? How does it, are there cures that need to be addressed? What's that like?
Well, I mean, you can't cure something until you know how it works. And right now, the big problem that we had, and we've actually now have solved this problem with Catherine Hoyle's lab. We now have the human imprintome. We have defined that through massive high throughput sequencing. And then the beauty of being at NC State is that there's massive numbers of computers and large storage facilities for analyzing and interrogating huge data sets, not because of biology, I don't think. I think it's primarily because of physicists, because they are used to interacting and getting big data sets that they need to analyze. But that means that we as biologists can also use the equipment and toys that they have to analyze the types of data and problems that we have. So this has become just a huge sequencing issue. And when you sequence, you can tell whether or not the methyl groups that are put onto the cytosine, you have cytosine guanine. So cytosine is on the left and guanine is on the right. On the other strand, which goes in the opposite direction, it's cytosine here, guanine here. I won't get into it, but it's the CG sites that are methylated on the cytosine. And when it's methylated, those cytosines are methylated, it tends to usually bring the DNA together. And as a consequence, the gene is shut down. And when those methyl groups, because the cytosines sit in the grooves also, and proteins can't get in and turn the genes on. So as a consequence, it tends to usually clamp them down and shut them off. And on the other hand, too, when you don't have the methyl groups, the genes are open and functional. So that's how these marks work. And so in effect, when you can determine whether a cytosine is methylated or not methylated through sequencing, which we can, then we can determine what's called the whole epigenome throughout every base in our body. And then what you'll have on these regions that are controlling the imprinting, one copy will be unmethylated over that region and the other will be methylated. So it's called differential methylation, and we can see that. So, but you're looking at 3 billion bases looking for about 1,000 sites that are about 100 to 1,000 bases long. So it's kind of like a little bit looking for needles in a haystack, but we found them.
So that, just talks right there about the complicated way that we even investigate where are these sites that we should be targeting. So my last question for you before we get to audience questions is, what's next? What is Dr. Hoya's team or your advisory team going to be doing in the next steps?
It's pretty clear, you already know. We now have the human imprintome, so we can now screen, and we already have done it in Alzheimer's disease, and it's incredibly interesting results we already have with that neurological problem. But the two that I'm really most interested in are autism and schizophrenia. They're diametrically opposed neurological disorders. Which of these imprinted genes are deregulated and what pathways are affected? The advantage of this is if there are some of these imprinted genes that are involved in this, we might be able to treat these, not by going in and doing gene therapy where you have to change mutations, but literally simply just opening up that copy that's being functionally shut down because of the imprinting process. And there's already people working on that at UNC. My next door neighbor, Ben Philpott, has found ways to open up silenced imprinted genes. So the ability to do therapy now, and with CRISPR, being able to go in and add things and stuff like this, the ability to actually go in and start treating some of these neurological disorders epigenetically, once you know which ones and where to treat, that's the next frontier. I mean, it's astonishing. What we wanted to do was determine the human. I didn't think I'd be able to see this happening, but it's done. the human imprintome is now done.
And that's fascinating. My husband was one of the programmers that worked on just the Human Genome Project, but now, and that wasn't that long ago that he was working on that. Yeah.
2000, 2001.
Yeah. So with all of that, I know that I don't see any questions in the chat box, but I'd love to hear from our audience if they have any questions about this work. So please type your questions in the chat box. But while we wait, I have one more question for you. And this is a little bit more about controversy. So you brought up the idea of potentially being able to open up the shutdown gene. I know I'm not using the right terms, but to be able to treat autism or schizophrenia or some other disease state. And my question is really about controversy around that, right? So we know that there are some challenges associated with having autism or challenges associated with having schizophrenia, but a lot of people have questions around, well, because it's a challenge, does it mean we should get rid of it completely? And my question to you is, have you interacted with people who are like, no, I would not want you to treat me in that way, or, oh, this is the ethics behind what you're doing? Can you speak to that at all?
I can speak to it, but I've never interacted with people like me. I like to interact with DNA. But I understand.
It's quieter, right?
No, but I understand your question. I mean, frankly, a number of people that are probably in science are over on the autistic spectrum, and people that are writers and artists and stuff tend to be a little bit more over towards the schizophrenic side of things. And what you're saying is that should we get rid of it? I don't think we'll ever get rid of it. We will never, because every generation they're recreated. But and you're never going to force people to do this, but some people are so severely affected that they might actually want to come in a little bit towards the norm rather than being way out on the edge. So I don't see the ethics issues as being severe because nobody's going to force people to do this, I don't think. But and I don't think you'll ever be able to totally eliminate those tendencies in people anyway. It just might make them be able to deal with life a little easier. So that's the way I think anyway, but I might be wrong, but I don't see that as being a real problem. But it's definitely an issue that needs to be talked about, because I thought about it myself. I mean, a lot of Edgar Allan Poe was this manic depressive. Yeah. Which is on the very edge of schizophrenia, and yet you look at the things that he wrote.
Yeah. Kendall, are you able to speak now, or do you want me to read? She's typing, so I'm guessing she wants me to read.
I can speak softly. So my question was, what is the main neurological disorder that you would like to see cured, and is epigenetics on the way to curing it?
Well, the main ones we probably work with originally are Alzheimer's and maybe autism and schizophrenia. But I think once the human imprintome we published, which we're writing right now, and this information is out, you'll see a lot of people screening all kinds of different disorders and biological disorders, not just neurological disorder, Prader-Willi, Angelman syndrome. These are all imprinting disorders that potentially now will be able to be cured. There we know where the imprint regulatory elements are, but there are other ones that we don't even suspect that they are actually due to imprinting dysregulation. It's possible that a critical gene in the Down syndrome critical region is also imprinted from our information. So is that one that is a simple gene change, a dosage change, would it affect and help? I don't know. Do you have to do this early in development, or can you wait till the developmental issues occur, and can you reverse it? Some of them I think you will be able to, others maybe not. Once you go through those critical developmental stages, it's a one-way street and you can't go back, even if you could get the gene regulation appropriate in adulthood. So these are all issues that will be addressed in the future, but until we had the human imprintome, we couldn't address them at all. Me personally, I mean, you're going to think this is crazy, but I would like to see psychopaths screen for imprinted gene dysregulation? I think it would be fascinating. I have no idea whether they're involved, but it would be an interesting question to assess. It's another behavioral issue.
Absolutely.
And it's worthwhile looking at. Now we can do it.
That's definitely an interesting.
It's weird, isn't it? But I think it's important.
Well, and I think, you know, that there are some potential positive implications of being able to identify the root causes.
Exactly. Yeah.
So Cindy asks, can you say more about the sense in which schizophrenia and autism are opposites? Can you clarify that?
Can I see her? Is she on?
That's Cindy. Yes, she's got the headphones on.
Yeah. She's the one that this is a very interesting question. Cindy, if you're really interested in it, I highly recommend that you go on Amazon and just plug in Imprinted Brain. It's pretty easy. It's a book, and it's written by Christopher Badcock. And he wrote this. It's an extension of why the phenomena of genomic imprinting evolved in the first place. Christopher Badcock himself, I don't think he would mind me saying this, has Asperger's syndrome. So he's over towards more, again, this incredible scientist. I mean, one of the smartest people I've ever met. And I think, you know, that's what we're getting back to in one of your original questions. Do you want to get rid of all these issues totally? Maybe not. But some people are almost incapacitated by these issues to the point where maybe they would want to be brought a little bit closer in towards norm so that it could function better. So he postulated, as an extension of David Haig's theory of why imprinting evolved, it's called the conflict theory. And he postulated, David Haig now postulated, imprinted genes evolved basically because there was a competition between males and females, adults, males and females, to control the amount of nutrition that the offspring extracts from the mother, with the father trying to maximize that extraction so that his offspring grow big and project his genetic information forward, whereas the mother trying to dampen it down because she knows that, for one, you would die in childbirth if there was the offspring became too big in utero. And the woman knows that she's related to all of her offspring, whereas the male doesn't necessarily know this. So that's a theory. So that means then that it made the prediction then that imprinting would be present only in placental mammals, because it is only once the placenta evolved that the father could get into the genetic game of controlling the amount of nutrition that the offspring extracts from the mother. So that means that any animal that lays eggs would not have imprinted genes. That made that prediction. Interestingly, mammals have members that lay eggs, the monotremes, and don't lay eggs, which are marsupials and the eutherians, which are the true placental mammals, of which we're members of. So we had a zoo in our freezer, and we went back evolutionary. This is before any sequence was known at all. So it was amazing that we were able to pull this off, but we did. And we demonstrated clearly that marsupials and eutherians at two imprinted loci were imprinted in marsupials and eutherians, and monotremes and birds were not imprinted, which is exactly what Haig's theory predicted. Now, it doesn't prove it, but remember, theories are like piñatas. They're for scientists to go and bang the hell out of them to see if something shatters and comes out of it, or whether it holds up. This one held up. So the extension of this is that this conflict does not have to end with childbirth. But if it's going to go forward after childbirth, then you're going to have to see, depending on the genes that are imprinted now, the individual demanding more nutrition, not just physiologically getting it through physiology changes in the placenta, but physically demanding more from the mother and more nutrition versus not. Once you use the word demand, it gets, the whole thing says that the battle went from the placenta to the brain. And that's where I'm saying that's the other place where they have the huge amount of imprinted genes expressed. Now, imprinted genes are not expressed uniformly throughout the brain. The paternally expressed imprinted genes are expressed primarily in the ancient brain, the hypothalamic regions, and the maternal ones, which tend to be anti-growth. The fathers are pro-growth, the mother's anti-growth. They're not distributed equally either throughout the brain. They're in the new brain, the neocortex. So there's a balance between this and this, a competition. And when that breaks down, what happens that as far as theory, and Christopher Babcock's theory is that it gives rise to two totally different biological behavioral disorders. When you have large brains, too much paternal, basically overall expression, it gives rise to what he says is it gives rise to autism. And on the other hand, when there's too much maternal break and the brain size actually is reduced in size, it gives rise to the antithesis of autism, which is schizophrenia. And every neurological disorder then, according to this theory, fits between these two extremes and are due to basically, not totally, but to a great degree, varying levels of paternal versus maternal being slightly out of balance, more here over to the autism, more here over towards the schizophrenia. Isn't it beautiful?
That is
if it's
really
true,
interesting.
I don't know.
Yeah.
But see, now that we know the human imprintome, we can actually start addressing this theory and seeing what these imprinted genes are doing in these two disorders. That's why I'm interested in these, what he calls diametrically opposed neurological disorders of schizophrenia and autism.
So
I don't know if we'll pull it off, but man, it's going to be exciting.
And I think that's why we love science so much, right? Is because we never know what's going to happen. I will say that we do have a copy of Imprinted Brain. Thank you, Danica, for sharing that with us, that at the library, that you can check that out. We're all about partnering with our library. And unfortunately, that is all the time we have for today, but I am sure Dr. Jirtle will be happy to answer more questions.
Just email me.
Yeah, just email him. And if you don't know his email address, you can look me up and I can email him for you. But we want to thank you again, Dr. Jirtle, for talking to us today. It has been a fantastic time. And I wanted to remind everyone that we broadcast every third Friday of the month at 2 p.m. And our next show in June, right? We're not there yet, will be Dr. Nilda Costco, an associate research professor in the College of Design and the director of the Natural Learning Initiative. She'll be discussing her research on preventing obesity by design. And you can find more information about this project on Wolfpack-Wellness website. So I am your host, Suzie Goodell, and our interns are Gethin Condren and Kendall Morrison. We will say a fond farewell to them as they have both graduated and are moving on to amazing things. Our webmaster is Truman Lee, and our research librarian is Cindy LaFleur. So until next time, be well, y'all.
And Cindy, thanks for that question.